Hello, everyone. My name is Dennis Scadding, and I'm the chair of the UK Semiochemistry Network. It's a real pleasure to welcome you all this afternoon to this meeting to hear Christina Buesching talking about her work with badgers. Christina did her MSc at the German Primate Research Center on mouse lemurs. She then moved to working on badgers in Oxford, doing a DPhil on olfactory communication in European badgers, meles. And she then became an adjunct professor at the University of British Columbia while still continuing her badger research. Christina's main focus is the role of olfaction in reproductive behaviour and the interactions between reproductive physiology, microbiota, and chemical signalling. But she's also very widely published on many other topics, including human-wildlife conflict and the illegal wildlife trade. And she's worked on a whole variety of species ranging from small mammals to koalas and giant pandas. We are extremely fortunate to have you talking to us today, Christina. Please go ahead and give us your talk.
Thank you very much for your kind words, Glenys, for inviting me, Krishna, Glenys, and Simon. And yes, I have actually a very long-standing relationship with the UKSN. As some of you might remember, I first came in 1997 and gave a short talk about my work on badgers. And that is how I met John Waterhouse, who became a long-term collaborator, and we worked on semiochemistry in badgers. And that really sparked my interest in the whole topic and made me realize how very important the role of olfaction and olfactory communication is in virtually every aspect of animal behavior, but especially also in mate choice and reproductive behavior. So maybe to set the scene a little bit, I have to, that we're all on the same page. I have to maybe repeat a few things which are fairly obvious, but what all animals want from life, of course, is essentially as many high quality descendants as possible. And that includes animals, includes humans, and of course, every other living being. But the way to get there, to have many high quality descendants, is different in each case. There are several things an animal can do to increase the number of descendants. For example, it is important to look after your own somatic fitness. So animals want to live very long, longevity. They want to be resistant against diseases, the immune system, and they want to obviously be in good somatic condition to survive themselves, but also to be able to care for their dependent offspring. And then, of course, they also have to think about their reproductive fitness. They want to have as many descendants in each litter and as many litters as possible in their life. They want to have very fit descendants. So all their babies should be fit enough to survive and should be well prepared to cope with life later on once they are mature themselves and can reproduce themselves. So well-prepared descendants to cope with the environment. But no one can have everything in the world, and therefore this comes with considerable fitness trade-offs. So looking after your own somatic fitness, investing in your own body fat, means that you can't invest as much energy into looking after your own babies, maybe in the case of females not producing as much milk as if you were prepared to not be that fat yourself. But these things are all very much dependent on various extrinsic and intrinsic factors. And really each individual animal has to make its individual decision. And what we often forget in the case of fitness trade-offs is that in addition to somatic fitness and reproductive fitness, there's actually a third component, and that is advertisement. If you are very fit and you invest a lot of energy into potential reproduction, that doesn't count for anything if you can't advertise that to potential mating partners and therefore get your own best mating partner and attract the most high quality mate. So advertisement is very important, and therefore basically this whole talk centers around the advertisement side of reproduction. Now, there are two categories of information which are important for animals to advertise in the context of reproduction. There's necessary information and more additional information. The necessary information is they want to advertise what species they are in or belong to. Very, very fundamental if you want to reproduce. Equally fundamental is what sex you are. And then, of course, what reproductive status. Are you already pregnant in the case of females, or are you still actively looking for a mating partner? Are you an immature animal which hasn't reached sexual maturity yet? Or are you actually on the market to find a mating partner? And then there's the additional information, such as somatic fitness and reproductive fitness, or expected reproductive fitness, individuality. It often counts who you are as an individual, and group membership, relationships, genetic ones and social ones, and so on. Now, as Glenn said, I've spent, well, basically 25 years looking at badgers and using badgers as a model. And they're actually almost the perfect model species to study olfactory communication because they're nocturnal. Therefore, olfactory signals are the primary means for intra and interspecific communication. Eyesight is not very good. Badgers make a lot of noise themselves when they're feeding, so vocal signals are not that useful actually. But olfaction is extremely good, and badgers made ample use of olfactory signals. They use virtually everything and anything you can think of. They use feces and urine and the secretion of specialized glands, such as the anal glands, which every carnivore has, and also subcaudal glands. And they use latrines. And that is where they deposit their feces, urine, and anal glands. And also when they walk around, they leave scent from their interdigital glands. And with the subcaudal gland, which is a specially evolved gland and unique amongst mammals, evolutionary, they mark objects and they mark conspecifics. So very important. From a more social point of view, just so that everyone knows basically what a badger is, badgers are considered primitively or facultatively social because there's very little evidence for true collaborative behavior. So they live together in groups, especially in southern England. They form large groups up to 30, 35 individuals. In other parts of their ranges are actually solitary or pair living. But despite living in these large social groups in England, there is virtually no evidence for collaborative behavior. So they're solitary hunters or foragers. They eat mostly earthworms, so not really true hunting, but still they're feeding on their own. And any social behavior, such as grooming, follows a tit-for-tat pattern, and they don't even warn each other of danger. They don't look after each other's young. So all very solitary lifestyle, actually. The only thing is they share a communal set, and a badger set is quite an extensive burrow system, and it encompasses up to 50, 60 different chambers where the animals sleep. And all these chambers are underground, up to 5-6 meters underground, depending on the geology. And they're connected with a very extensive tunnel system. And that is what the badgers in the same group, all help each other, if you like, collaborate on. They actually all maintain this badger set and dig more tunnels and chambers, but also change bedding material, collect bedding material, straw and leaves and so on. Now, we also need to know a little bit about the badger reproductive biology. And the first thing you will notice if you watch a group of badgers is that badgers display no obvious sexual dimorphism visually. Part of that is, of course, down to them being nocturnal. So seeing another badger wouldn't show them if they're males or female, because you can't see very far. But still, there's no obvious sexual dimorphism in badgers, and it goes down to the millimeter. If you measure hundreds, thousands of badgers, as we have done, there's, I think, 1.7 or something millimeter difference. Males are slightly bigger than females on average. Badgers have a highly promiscuous mating system, and they're polygynandrous. So literally everyone mates with everybody. They have a main mating season. In England, it's restricted to January, February, so mid-January to end of February, and that coincides with the postpartum estrus. Because the last point, females give birth in mid-February, around Valentine's Day. So around now, there are about 2-week-old baby badgers on the ground. They're born very altricial, blind, and ear canal isn't open. And the females are mating. And per litter, there are about 1 to 4 cubs. However, only about 1/4 to 1/3 of the females in any given population give birth to cubs each year. And that, of course, is already a sign of high intraspecific reproductive suppression. Now, interestingly, badgers mate in mid-February, but they're polyestrous, so they mate, they become pregnant, the eggs are fertilized, but they undergo delayed implantation. The fertilized egg does not implant into the uterus wall, but the blastocyst remains suspended in suspended animation in the uterus up to 9 months. And then only around the shortest day of the year, around 21st of December, the blastocyst actually implants and proper pregnancy starts with morning sickness and everything else likely. And the badgers are pregnant in our sense of the word. And during that delayed implantation, badgers can undergo additional estrus cycles and can mate successfully, and that leads to or results in superfetation and superfecundation, and can lead to multiple paternity within the same litter, because not all the cubs are actually fathered at the same time. So determining the reproductive status of badgers is actually quite difficult, and we've done, I think, about 10 publications on how to measure if a badger is in reproductive condition and pregnant and so on. You can look at the morphological indicators, length of the teats to see recent lactation, descent of testes and so on. You can look at sex steroid levels in the plasma, but also in feces and urine. Of course, sex steroids get metabolized and excreted. Or you can even look in fur, in keratin structures, so hair and claw, and get a longer term picture of the hormonal status over the last 2 months or so. Each of these methods has advantages, and each of them has disadvantages. And it really depends on what you want to look at, which method is the most important or suitable one. So now that you've got the basis about badgerology, we'll turn back to scent marking. And basically, as you all know, there are 2 fundamental categories of scent marks. One are excretions, so metabolic byproducts, urine and feces, and to some degree saliva. And the other one is specialized gland secretions, which have evolved purely for the function of scent marking. So feces and urine as metabolic byproducts, they obviously encode things like food, feeding activity, how much water the animal has been drinking, which might be important in desert animals or during times of drought. And because anything in the body has to be metabolized, they also are the pathways to excrete sex steroids. So they include things like sex and reproductive status, whereas glandular secretions are a lot more open to really be encoding proper information which is evolved specifically for communication. And therefore, they are also more likely to be suitable as honest fitness advertisement, because only an animal which wants and can use surplus energy into in secretions can actually, is fit enough, very obviously, and therefore can be considered fit and can advertise its fitness honestly. But the information content is very much open to many different factors affecting it. Broadly, we can distinguish these or classify these two different things as cues and signals. So cues would be feces and urine, sort of things which occur naturally. Signals are purposefully left scent marks. However, badgers don't read the textbooks, and therefore they actually use the cues of urine and feces very much as signals. They use them, have modified their behavior to defecate and urinate in very clear signaling locations. They use latrines, and these latrines can tell us an awful lot. We've been studying them for over, well, actually now 50 or 60 years. And over that time, we see that latrine locations remain relatively stable. So even in a study site like Whiten, which is 1,000 acres, the latrines which Hans Krug found in the 70s are still in the same place in many cases, now 50 years later. So that again shows us that badgers seem to really want to know where those latrines are and that they're important in communications context. There are two types of latrines, hinterland latrines as well as border latrines. And the hinterland latrines are used only by one social group, whereas the border latrines are used by all neighboring groups. Now, how do we know this? Badgers love peanuts, and therefore we can feed badgers with peanuts and mix those peanuts with different colored plastic beads. So we feed one group of green beads, the next group with red beads, and the third group with yellow beads. And so that the badgers don't just pick out the peanuts, we stick the plastic beads with treacle to the peanuts. So we mix our bucket of peanuts and plastic beads with treacle. And then we feed each set with a different color. And then we do that for 2 weeks, and then we go surveying for about 2 weeks, and we find those lovely colored feces. And since we need to do that when the latrines are most active and also vegetation is lowest, Easter is a fantastic time to survey for latrines. So Easter egg hunt has a completely different meaning for any badgerologist. But if you survey the woods, then you see that some latrines are used only by the red fed group, others are used by red and blue and purple, and therefore we can see hinterland latrines and border latrines. And if we put those colored latrines on a map, then you come up with that picture in the middle of the screen there, sort of a nicely colored map of Whiten, where each area of Whiten Woods is filled in with a different color. So it looks like the purple badgers stay in the purple area, the yellow badgers stay in the yellow area, and so on. Very nice visual indicator for us visual animals that something is going on with latrines and that they are important to badgers. And if we go back to the field and watch what badgers do at the latrine sites, then we see that they indeed spend an awful lot of time at latrines. Individual badgers often travel very long distances to use specific latrines, to the point where they might feed in the southeastern corner of their territory and then travel 2 kilometers to the northwestern corner of the territory simply to sniff the latrine and use the latrine. And then they go back to their southeastern corner and continue eating. So it is a very clear signal that latrines are incredibly important to badgers. Interestingly, they show very little aggressive behavior at latrines. So normally if a badger from group A walks into the territory of group B, or even worse, into the territory of group C, and the badgers meet each other, then there is some kind of regression. Usually they get away with growling and snarling and maybe a little bit of sort of air biting and showing their big canines, but sometimes they actually end up really going for each other and fighting and rolling around on the ground and seriously injuring each other. At latrines, badgers can meet and they don't take any notice of each other and simply pretend the other animal is not there, and they keep sniffing the feces. Interestingly, however, and that is contrary to our lovely colored map of the study site, badgers seem to not appear to respect latrine sites as boundaries. So while we made this nice distinct map of purple next to yellow with no overlap, the badgers don't know this. As I said, they don't read the textbooks. Badgers from the purple side go to the purple-yellow latrine, defecate, and then they march straight on into the yellow territory, where they do not defecate, but they walk around and eat and everything else. And then when they need to go to the loo again, they go back to their purple latrine and defecate. But in addition to using feces, if you watch latrines, you see that badgers scent mark extensively at these sites too, with other things like urine and glandular secretions, so subcaudal, anal, and interdigital glands. And yeah, they do sniff every scent mark very, very carefully. So if we just reiterate this point, if we look at the latrine marks or latrine colored site of the study site, then we see the underlying nice map. If we look at badger visits to latrines, then we come up with map A. So badgers sort of visit all the latrines which are in their vicinity. But on the map B on the right-hand side, that is a radio tracking map. And we see that the badgers from the, I can't point on the screen, but in the big area on the left, the ochre, beige, brown, yellow, purple, and the light blue badgers, they all move around in that whole area, although they defecate only in their own little area. Now, badgers sniff the feces. So that shows us that it is very important to know what's in there for them, and therefore for us as scientists. And we said already they're metabolic byproducts. So hormones are very clearly in feces and in urine. And they encode sex, age, reproductive status, and seasonality, because badgers are highly seasonal breeders. So they have that one main mating season, January, February. And yeah, therefore it varies with season. However, if we look at the hormones in urine, then in the top graph, you see testosterone in plasma and in urine. The testosterone in urine is the solid line, in plasma, the spotted line. And you can see that by and large, it's not seasonal profile, it's actually different individuals on the x-axis. But by and large, individuals which have high testosterone levels in urine also have high testosterone levels in plasma and vice versa. Underneath, you have the same type of graph for females, estrogen in plasma and urine, and you see that there's very, very little connection between the two profiles. So urine is maybe not the best way for us to look at, and that means badgers are probably not as interested in urine as they are in feces, where the can't think of the word, is much stronger, the relationship. However, even in urine, we can see that cubs, the green circles, and adults, the blue circles, can be very clearly distinguished simply on the sex steroid levels, and that is true in males and in females. So even if a badger sniffing urine can't figure out if it's a female in estrus, he can still figure out that it's an adult female and therefore a potential mating partner. So, of course, much more important, as I kept saying, are the gland secretions. And badgers have, as carnivores, anal glands. So all carnivores have paired anal glands. And in addition, they've evolved the specific subcaudal gland, which is actually a skin pouch lined with apocrine and sebaceous cells. And that is unique to badgers, to the genus Melis. And therefore, it's an indicator that this really is very important in badger behavior. Both sexes have both glands. Both glands show very big differences in color, which of course indicate differences in chemical composition, and also volume varies a lot between individuals and seasonal and age classes. So the volume, of course, is a bit of an indicator for how much each individual invests in producing the secretion. So if we look at this profile, you can just see this is for subcaudal gland secretion. Seasons are on the x-axis and color rank, so from light to light being 0 to dark, basically black being 6. You can see that there is a nice seasonal profile. Secretions in males and females are always lightest in spring and darkest in autumn. And the same is true for volume. Animals have the most secretion in spring and the least secretion in autumn. So again, seasonal pattern shows that this is important. But what actually is important to badgers is not the color, they're colorblind, but it is the smell. And to us, people often ask me in the past, what does subcaudal gland secretion smell like? I can't say. I once gave a talk at the UKSN network at a specific forum, which I didn't look at who was actually in that. And afterwards, people asked me, so can you describe this? Does it smell musky or not? And it turned out to be a forum for perfume producers. I can assure you, while I can't describe the smell of subcaudal gland secretion, I don't think it's something you want to anoint yourself as humans. It is rather rancid smelling, and we really have to look at the chemical composition, either with solid phase micro-extraction or with solvent extraction, to study the composition. We have used both methods in both secretions. And we find that anal gland secretion contains a very high number of quite short-chained volatile organic components, and it is very pungent. It is quite an immediate smell. If a badger sets off its anal gland secretions, you can smell it. It's very many aromatic compounds, and then, of course, fatty acids and hydrocarbons. Anal gland secretion, the composition varies very obviously very significantly with season. So this is spring, green, autumn, and summer. We didn't have winter samples, but the few winter samples we had were separate again, very separate actually, main mating season. It varies or differs between the sexes, blue for the boys, pink for the girls. It encodes age, so the open green circles are the cubs, smell very differently to males, to adults, the blue circles, and importantly, it encodes individuality. And each symbol on this graph is a different individual, and all of these individuals have 3 to 4 different samples analyzed, and they are collected during different seasons of the year. So spring, summer, autumn, and winter. And you can see that even during different seasons of the year, samples collected from the same individual smell more similar to that individual than to other same season samples. And importantly, if you think of the territorial system or the spatial system of badgers, anal gland secretion also encodes group memberships. So animals from the same social group smell very differently to animals from different social groups. The same is true for subcaudal gland secretions. They contain considerably longer chained fatty acids, so they're not as volatile, and they have a lot of protein in them and water, but mostly long chained fatty acids, which make them much more suitable for a long-term signal because they're not as volatile. But again, subcaudal gland secretion, the composition varies with season, with individuality and group membership, also age, but it encodes also other fitness-related parameters such as body condition, health, and hormonal status. So subcaudal gland secretion seems to be very important indeed in badger behavior. Now, if you think back to the most important thing an animal has to encode in its signals when it wants to advertise in a reproductive context, it has to encode species. Are you the same species or are you a different species? And therefore we looked at badgers and compared samples from different areas between England and Tokyo, so Bulgaria, Kochi, and Russia, and so on. And Chinese badgers are actually not on this graph, but should be. And of course, Meles anakuma is now a different species. At the time when we did this analysis, it was actually still a subspecies. But very clearly, animals, badgers from different regions smell differently to each other. So they do encode also species-specific smell. However, especially in the subcaudal gland secretion, we have a lot of protein. And that is something I'm just trying to get a handle on and have a collaboration with Sunita Janssenswillen in Belgium. Proteins are just being recognized really over the last 1 or 2 decades, how very important they are in olfactory communication. They're much longer lasting than volatiles. And of course, they're gene encoded and they bind directly in many cases to the vomeronasal organ. So they're specifically targeting or co-evolved with the vomeronasal organ with the scent organ. And the role of proteins can either be as a chemical signal in itself, like dancing in mice. Jane knows all about that. But they can also be functioning as a carrier for odorants, for volatile organic compounds. But usually if they do function as a carrier, then they slow down the release of these odorants, making the signal smell longer, lasting longer. Now, in badgers, we have started to look at the protein content, and we do see indeed that in the top row of the 4 graphs is summer, bottom is autumn. You can see that there are differences in concentrations overall in protein content, but also in different seasonal context. And on the left is males, on the right is females, and again you can see that there are very distinct sex differences. However, proteins especially, but also fatty acids are quite expensive to produce. So can the cost of the secretion actually indicate the fitness of a badger, sort of also give an indicator of somatic fitness? And that is what my student Tanisha Allen looked at. And she came up with the idea of using a bomb calorimeter to measure the overall cost of subcaudal gland secretions. And that really functions the same as wanting to find out how many calories are in a piece of chocolate. You burn the whole set volume of subcaudal gland secretion and measure how much energy gets released by doing so. And if you do that, then you see that there is considerable inter-individual variation. And on the graph, which is very pale, I realize, on the left, you've got females, sort of the reddish graph. On the right, you've got the males. And you can see that in females, there is a very considerable inter-individual variation, whereas on the males, so huge standard deviation. On the males, basically all males have kind of the same level of calories in secretion. So overall, calorific value of male secretion is slightly higher than in females, but standard deviation is much higher in females than in males. And now we're looking at if badgers can actually or do use these costly secretions as honest signal advertisement. And the test, of course, is if badgers coming across different secretions of different calorific value react differently to them. And it turns out they do. On average, badgers sniff costly secretion longer and more intensively than secretion of lower calorific value. However, you will realize that at the moment, this is only an indicator, and we actually don't know what makes these secretions so costly or high calorific value. Is it the proteins, is it the fat, or is it something else? So this is just an indicator at the moment, but we're starting to look at that. So, of course, there are many other factors affecting the information content of secretions. We've sort of touched on various methodological differences, SBME compared to extraction, solid phase extraction, thermal desorption, and so on. Your GCMS protocol, but also your statistical analysis has huge implications on your own conclusions on the results. But overall, there is also a lot the animal, the individual itself can do to vary the scent of secretion, scent of the secretions. And the most important thing, of course, is the primary gland products, what it produces and puts into the secretion, in what concentrations it secretes various different components. But then there are also things like impurities and contamination, and the bacteria which metabolize the primary gland products. And it is those two last things we want to look at first, because the primary gland products we've talked about, they depend on individuality and fitness, sex steroids, stress hormones, food, time of the year, and so on. But they are always being metabolized by the bacteria in the secretion. No secretion is sterile, and every secretion therefore contains bacteria. And it's those bacteria which metabolize the primary gland products, which are actually responsible for the specific odor we then measure with our GCMS. And the bacterial fauna is sort of incredibly important and incredibly complex. I don't expect you to memorize this slide. This is just a fraction of all the different bacteria we have found in badgers of caudal secretion. So very, very complex fauna. And some bacteria are vastly more proliferant than others. But like every bacterial infection, the badgers actually have to actively manage their bacteria in their secretion. Otherwise, they end up with a scent they don't actually want to have when they scent mark. So it is very much a mutualistic, symbiotic relationship between the gland, the badger, and the bacteria. And the badger can change its growth conditions for the bacteria, and the bacteria therefore can hopefully generate the odor which is desired by the badger individual. But how can an individual badger manage this balance between resident and transient or infection microflora? So something they don't want to have in there, because the behavior, of course, is vital in this context. Badgers scent mark with the subcaudal secretion, especially by walking along and sitting down on the substrate they want to scent mark for a fraction of a second, and then they walk on. And when they sit down, they leave a bit of secretion, but they pick up a lot of bacteria from the substrate they're marking. And they can mark either inanimate objects, so the soil, plants, the set, and so on, or latrines, or they can mark each other, mutual other marking. And both of these marking behaviors lead to floral infections, so the pickup of bacteria, which then need to be managed. And the badger can, of course, decide to scent mark a lot in a desired area with desired bacteria and avoid others areas. But more importantly, and probably more precisely, it can manage the bacteria by managing its own physiology and the growth conditions for bacteria in the gland. Most importantly, it can manage the pH of the gland, it can manage the temperature to some degree, and it can produce things like, for example, bactericides, specifically targeted poisons to kill special bacteria. So if we look at the behavior, then we see that object marking does indeed lead to the pickup of various soil bacteria, which we then find in the secretion. It leads to pickup from feces, especially E. coli, when the badger marks at latrine sites, but also plant-specific bacteria. So there's really a very big chunk of the bacteria we found in subcaudal gland secretion comes simply from object marking behavior of the environment, whereas others are very clearly from scent marking conspecifics, so much more mammal-based bacteria. Oops. Okay. Yeah, okay, sorry. And now the badger wants to make sure that it can control which bacteria do well and which ones do not so well. For example, with its own body temperature. So there's temperature in the gland. You remember, it's a skin pouch which goes sort of 2 centimeters from the outside underneath the anus, which means underneath the end gut into the badger's, well, it's a skin fold, but it's in very close contact basically with the gut, which is very well, has a lot of blood circulation. And therefore the temperature within the skin pouch of the subcaudal gland secretion is body temperature, 38 to 38.5 degrees Celsius. Whereas most soil-based bacteria, of course, have a much lower optimum growth temperature range. So they're already disadvantaged. The pH of the secretion is actually very acidic. It's about 4.0. It can go up to 5.8, but that is only in some cases. Our skin or dogs are much higher pH. But we've seen in humans and in dogs, and we haven't done that in badgers yet, that there are very distinct age and sex differences, and also race or breed differences in the pH and of the skin, but therefore also likely of sweat secretions and so on in humans and dogs. And we believe that this is also true in badgers, something we need to look at. But of course, badgers could use either active proton pumps or passive ways like lactic acid to moderate the pH of the secretion. And again, acidity inhibits many of the harmful bacteria like E. coli, Staphylococcus, and so on. And then we have found quite a wide range of proven bactericides in the secretion, namely the long-chained fatty acids, octanoic, dodecanoic, and linoleic acid. So badgers secrete them in very high concentrations, partly probably as an olfactory signal, but partly maybe also to moderate and control their microbiome. But of course, all this is only as good as if the badgers say, yes, our results are true and actually true indicators of what the badgers are interested in. And therefore, we have to take our observations and chemical analysis, compound identification back to the field and actually do scent playback experiments. There are no shortcuts. And just to remind you, we can look at latrines, object marking, and allo marking. And there's very distinct activity change between the mating and cub rearing season and then the reproductive quiescent time of the year in summer. And if we look, so that was natural scent marking behavior. If we do that with scent playback experiments, where we put secretion at a latrine or at a set and monitor with a wildlife camera what happens when a badger comes along, then we can see that badgers are indeed very interested in our secretions. So where the adult there is sniffing behind the cub is where the secretion is. On the left, where the cub sort of looks to, there is our control sample, water, and no one is interested in that. But we can see that females actually bring their cubs from a very early age on to the latrines and show them this is what you need to read to know what's going on in the world. They scent mark, so every badger which is sniffing is also scent marking, and this animal does both at the same time. And they often meet at latrines, so you can see the defecating badger on the right, he's also sniffing, and the badger on the left is already on his way to sniff the feces, basically, right off the production line. So we can see simply by watching latrines and scent playback secretions that badgers are very interested in this. They do indeed respond differently to group and individual specific information encoded in both secretions, anal gland and subcaudal gland secretions. They moderate the response to these secretions according to familiarity, so same group, neighboring group versus strangers, and also the perceived coinciding threat level. So they are interested in their neighbors, but they're not scared by them. Whereas if you put secretion from an unfamiliar animal from the other side of the woods out, then they are not just interested, they're actually scared. Then they go into overdrive and start digging up the soil, scent marking everywhere, and a lot more animals start coming. And badgers moderate their response to all secretions, including urine, very importantly, also according to the encoded information, so sex and reproductive status, in relation to their own physiological status. So a male in reproductive condition will react differently to male secretions, depending if these males are, the donor males are in reproductive condition or not, and he will react very differently to female secretions than to male secretions, but also, again, is the donor female in estrus or not? So you can really see that this is a very important way of advertising and communicating reproductive context. And we can only liken it to our own Facebook page, which is more of a written or visual signal, but badgers really use scent marks to advertise themselves, so their individuality and fitness-related parameters, their own social group, who their friends are and who their family is, their home range, where they can be found. Doesn't pay if you just say, hey, I'm looking for a boyfriend and I'm so very attractive, but no one knows where you live. Doesn't work. You need to include information where you can be found. And in the case of scent marks, you also need to or want to include information how old the scent mark is, because of course it's a long-term signal. So it isn't enough to just leave a scent mark and say this female is in estrus right now. No, she needs to also say this is now, and the scent mark needs to degrade in a set pattern so that a male coming across the scent mark can say, oh, this was 2 days ago, or it is still current information. So I hope I could persuade you that we know actually quite a lot about badgers and reproductive advertisement in olfactory signals. However, there are, of course, very important gaps still. What we don't know is if scent profiles are hereditary in any way. Proteomics seem to indicate that they are at least individual specific, so they're gene encoded. Primary gland products might well be hereditary. We don't know how MHC affects scent in badgers, very big hot topic in many other species. We have some indication that it affects in badgers, scent in badgers too, but interestingly, badgers do not select mating partners for maximum MHC diversity, like many other species, but actually for similarity. So that makes it a little bit more difficult to see the effects of MHC on mate choice. We don't know how diseases, especially STDs, affect scent and olfactory mate choice. Chlamydia, we know, smells very strongly and off-putting. Is that the same in badgers? You want to avoid mating with someone who's got an STD. And therefore, also simply, how does the genital microbiome, the healthy genital microbiome, which is individual specific, how does that affect mate choice in also healthy animals? And we don't actually have any idea yet what the olfactory and semiochemical fitness consequences of differing gut microbiota are in badgers. And again, we're just realizing over the last decade or so how very important the gut microbiome actually is in maintaining somatic fitness, even in humans and all those things. So it would be so much easier if the animals could talk and we could learn the languages, but we could discuss these things directly. This way, we're simply limited to looking at the animals and looking at what they're communicating with the nose.